Hi, everybody. Welcome back. In the last video, we discussed how the number of molecules inside cells, at least for the molecules of particular interest, proteins and RNAs of various genes, may well be present in very, very small quantities, with RNAs present only in the countable on your hand numbers, and proteins probably numbering only in the sort of maybe thousands, and in some case only hundreds of molecules. And under these conditions, mass action kinetics breaks down. In this video, we're going to start discussing a little bit about what we need to think about thinking about biochemical reactions in this very sort of microscopic level when molecule numbers are small. Let me begin by switching over to the slides. So let's think a little bit about chemical reactions at the submicroscopic level, at the molecular level. Well, at this level, molecules are constantly moving around, bouncing around, bumping into each other in what is really an effectively random pattern. Now, there may be underlying rules governing that motion, but there are so many collisions happening, so many changes of directions, that keeping track of the rules governing that is essentially impossible, and the motions are effectively random, with molecules moving all about in different directions. Now, for many chemical reactions, in order for a reaction to occur, molecules must collide in a certain way. They need to kind of bump into each other, and they need to bump into each other in the right orientation. This is absolutely the case for any reaction involving two molecules, whether that's a bimolecular chemical reaction or a binding reaction. But it also may well be required for many reactions that are kind of even fundamentally unimolecular. They may need to get a bump of energy from somewhere in order for the reactions to happen. And it can also be collision dependent. Right? Molecules that are present in higher concentrations, that is, they're in higher densities, this will naturally collide more frequently. This makes sense. The higher concentrations there are of particular molecules, the more likely they are to be undergoing collisions. And so we get a concentration dependence on how often there are collisions as a necessity for chemical reactions. Some reactions occur easily. That is, almost any collision that occurs, a reaction will happen. Other reactions require a very special kind of collision, that the molecules have to come in and really bump each other in exactly the right orientation, with exactly the right kind of relative momenta for a reaction to happen. And so collisions lead to reactions more infrequently in that case. And all of this underlies the basics of chemical reactions. If you have more collisions, you have faster reactions. If you have easier reactions from collisions, you have faster reactions. So we can sort of think about this. We could zoom in to a tiny region inside a cell, and if we could watch the molecules inside there, what would we see? Well, we'd see molecules zipping through the region, sometimes just shooting through and not making any interactions and exiting. Sometimes we'd see two molecules bang into each other and change direction. Sometimes we'd see two molecules collide and lead to a reaction. And if we were looking at this, we just sort of focus, this would appear completely random. The directions of the molecules as they shoot into this little region would seem to be random. The timing between appearance of particles and between collisions would all seem random. Which collisions lead to a reaction, so productive collisions, as opposed to just leading to changes of direction. There wouldn't be any pattern discernible in any of this, any sort of rules discernible in this. It would all fundamentally appear to be random. If that's what's happening, how does mass action kinetics come into this? Mass action kinetics doesn't have any randomness to it. It's all governed by very, very clear rules. Well, in macroscopic kinetics, we are essentially averaging over billions or trillions or even more moles worth of individual reactions. And the law of large numbers makes this average very predictable. The law of large numbers is a statistical law that basically says that if you make an observation many times, if you observe some variable many times, the average that you observe, once you hit a certain number of interactions, a large number, the average is very accurate. Even if you have a fairly broad distribution, even if there's a lot of sort of random components to building up each individual observation, when you average over a large number, that average is very stable. And in macroscopic experiments, you're looking at an average. And you're looking at an average over a very, very large number. And so everything looks very deterministic, very easily understandable. Now, there is still a relationship. The overall reaction rate that we see is directly proportional to the probability of a reaction happening. That is, what's the probability of those individual, if we zoom down and we say how frequently do we see a reaction? In other words, if we watch for a certain amount of time, what was the probability that we would see a reaction occurring? The overall reaction rate in mass action kinetics comes directly from that. And with a higher probability of a reaction, a higher frequency of reaction, resulting in a higher rate. The concentration dependence that we see in the rate laws, that unimolecular reactions depend only on that individual concentration, that bimolecular reactions depend on the square, and so on. That concentration dependence comes directly from the concentration dependence of collision frequency. That's why there's that concentration dependence. The rate constants in the rate laws come from the probability that a collision actually leads to a reaction. So reactions where that have what we call a high reaction cross section, that is many different sort of variations in direction of collisions and energies of collisions all lead to a reaction, lead to higher rate constants. Reactions that require a very, very specific type of collision lead to lower rate constants. Right. Mass action kinetics depends on the law of large numbers to average out this fundamental random behavior in molecular interactions. And so, unsurprisingly, when we go down to a small number of reactions, mass action kinetics doesn't work anymore because the law of large numbers no longer applies. Right? It's called the law of large numbers for good reason. It doesn't work with small numbers. when we do have this case, when we have small numbers of individual observations, small numbers of reactions, the average that we observe might be different from case to case. We haven't reached the population sizes needed to have a consistent average from experiment to experiment. And so we will see the average changing depending on the details, right? And this might be important. Right? To model problems of this, right? To describe in a accurate way the behavior of systems that are in this small number conditions, we need a model that is able to consider the underlying random nature of chemistry. So let's explore this a little bit. We're not really going to develop the model in detail in this video. Let's just introduce a simple biochemical system with this randomness, this stochasticity. Stochastic is a word you're going to hear a lot over the next few videos. Really, it's a synonym for random. It means sort of occurring based on random chance. So let's think about the production of a protein, call it X, from a constant concentration of mRNA. Right? You might imagine this being, for example, an in vitro experiment or an experiment outside of a cell in which a fixed amount of RNA, let's say amount, not about, of RNA, ribosomes, tRNA, and everything have been added. So you have a fixed background, and then the amount of the protein is being measured. Right? And we'll include degradation in this model also. The mass action inspired model, the sort of model that we've been working with so far in the class for a system like this, would have a single equation. We wouldn't have an equation for RNA because it's fixed, and really a single variable, the rate of change of protein concentration, a synthesis term that depends on that RNA concentration, but that's a constant number, and a degradation term that depends on the amount of protein. There's only one equation, two processes, synthesis and degradation. This is what is sometimes referred to as a birth-death process. There's one thing that can make them all, make something, and another process that can remove it. Right? Synthesis, degradation, birth, death. This type of system has been studied fairly extensively from the stochastic modeling perspective. And the equation that that corresponds to, if we simplify things with simpler notation, is one with a well-defined analytical solution. It basically corresponds to a differential equation, dx dt, that has a positive constant and a linear negative term. This differential equation has a well-defined analytical solution of x as a function of time being a constant, given by the ratio of the synthesis and degradation constant, minus another constant multiplied by e to the negative mu t, or that's the mu is degradation constant. Okay. And x0 here is the amount of protein that or the starting initial value that we always need when we integrate a problem. Right? If we think about what this solution, this general solution describes, we have a constant and then minus an exponentially decaying term. This describes an exponential approach to a steady state. When t is 0, this will be e to the negative mu t will be 1, which means that the alpha mu and the the alpha mu's cancel out, and we're left with our initial condition. When mu of t, when t is large, e to the negative, a very large number, approaches 0, which means this second term disappears, and we approach this alpha over mu. Plugging the equation of the notation for the problem into this model, our alpha is omega times RNA concentration. The mu is chi x, and x is, of course, protein concentration. And so the solution is given here, that the concentration of protein with respect to time has this constant minus this constant multiplied by e to the, there should be a negative there, apologies. So I'll put that in. Should be a negative in the exponential there, e to the negative chi x t. And of course, this has a, as t goes to infinity, once again, this second term disappears, leading us to that. It has a steady state, an equilibrium value defined by this. Now, what happens at the molecular level for this? That was the ODE model. At this sort of low-level molecular model, if we think about a very small portion of time, a tiny little bit of time, we think about what might happen. A new molecule might get made, or it might not. An existing molecule of protein may degrade, or it may not. Maybe more than one of these things will happen. Maybe neither will happen. These first two, whether a protein gets made, whether a molecule degrades, these are random events. They're chemical reactions driven by these sort of underlying random interactions, and the probabilities of those are going to be proportional to the rates. The probability that a protein is made is going to be proportional to omega x times the RNA concentration, where RNA concentration is fixed in this model. The probability that a protein degrades will be proportional to chi x times the protein concentration. Protein concentration is not fixed. The second two possibilities, whether more than one of these events occur or whether no event occurs, can actually be derived from these first two. So it's and if we consider a very short time period, we should be able to find a situation, where the probability that more than one thing happens is vanishingly small, is very, very tiny. And this is going to be an important point that we get into later when we sort of develop these models in more detail. If we build a simulation that kind of captures this, that has a random probability that proteins might be made or proteins might be degrade proportional to these rate constants, what we actually get in a simulation is something that kind of looks here in the blue jagged trace, is a stochastic simulation of this system. And the deterministic, the solution for the ODE is shown in the dashed red line. And what we see here is this particular stochastic solution. You see that sometimes molecules are made and the number goes up. Sometimes molecules degrade and they go down. Sometimes molecules grow up for a while, stay still flat for a while, degrade, quickly grow again. And we see they're going up and down, up and down, up and down. The trend follows the general path of the deterministic solution. And this is really what we expect to see in some sense, right? The deterministic solution doesn't come from nothing. it. If we were to repeat this simulation, every time we would do it, the stochastic simulation, every time we would do it, we would get a different trace of up and down concentrations. It would always roughly follow this line, but the details would be different in every case. If we average those out in the right way, we would get this line as the average. One of the things that I think is very important to sort of notice in this is that if we think about steady state, this time where the average number of molecules, the ODE, the mass action kinetic based description of the system would say there's no change, there is actually still a lot of fluctuation. We can go as well as the deterministic solution levels off at 10. We find sometimes as many as maybe 16 or 17 molecules. We go down to as low as 4 molecules. Very, very big swings in the overall numbers, even under steady state. Right? And this can be very significant, or maybe very significant, for biological function under conditions where this stochastic behavior is important. So in summary, what we've seen, we've discussed how chemical reactions are fundamentally driven by random collisions, and that the mass action kinetic models that we've discussed so far are macroscopic models that describe the average behavior of large collections of molecules undergoing these random collisions. And it's the fact that we have large numbers that lets the models be simple and deterministic. The macroscopic rates are directly related to the probability of individual reactions happening, but there's this averaging that goes on in moving to mass action. When the number of molecules involved is small, though, the deviations from the average that we see in an individual trajectory or in an individual set of observations can be very big, even sort of on the same order as the numbers we're measuring. And that's kind of really one of the essential points. This gives you a little flavor of where the underlying ideas of the types of random interactions that lead to chemical reactions and the connection to mass action kinetics. Over the next few videos, we're going to dig into this more deeply, develop the tools to robustly describe and simulate biochemical systems with a stochastic chemical theory. Before we do that, though, in the video directly after this, we're going to do a little bit of a review of probability theory, or introduction to probability theory, depending on your background, just to set the page to get all of the language in place that we need to develop these models. Thank you for listening. Until I see you next time, take care and be well.